Welcome to our exploration of bacterial physiology. Bacteria are among the most fascinating and diverse organisms on Earth. They thrive in environments ranging from deep oceans to human bodies. Understanding their physiology helps us appreciate how these tiny life forms survive, adapt, and impact our world. In this video, we'll dive into the core aspects of bacterial physiology, from their structure to their metabolic processes. Let's begin by setting the stage for what makes bacteria unique and why their physiology is so important in science and medicine. Bacterial cells are simple yet highly efficient. Unlike eukaryotic cells, bacteria lack a nucleus and membrane-bound organelles. Their genetic material is found in a single circular chromosome located in the nucleoid region. The cell wall made of peptidoglycan provides structural support and protection. Some bacteria also have an outer membrane, capsule, or flagella, which help them move and evade immune responses. These structural features are crucial for their survival and adaptability in various environments. Bacterial cell wall is a defining feature. It's primarily composed of peptidoglycan, a mesh-like polymer that gives the cell its shape and rigidity. Gram-positive bacteria have thick peptidoglycan layers, while gram-negative bacteria have thinner layers and an additional outer membrane. This difference is important for antibiotic susceptibility and staining techniques. The cell wall not only protects bacteria from environmental stress, but also plays a role in their interaction with hosts and other microbes. Bacterial membranes are vital for controlling what enters and exits the cell. The plasma membrane is made of a phospholipid bilayer with embedded proteins. These proteins facilitate transport, energy generation, and communication. In gram-negative bacteria, the outer membrane contains lipopolysaccharides, which can trigger immune responses. Membrane functions are central to bacterial survival, allowing them to maintain homeostasis and respond to environmental changes. Bacteria carry their genetic information in a single circular chromosome. Some also possess plasmids, small circular DNA molecules that can be transferred between cells. Plasmids often carry genes for antibiotic resistance or virulence. The nucleoid region is where the chromosome is located, and it's not separated by a membrane. This arrangement allows bacteria to rapidly replicate and adapt, making them highly efficient at evolving in response to environmental pressure. Bacterial metabolism is incredibly diverse. Some bacteria are autotrophs, producing their own food from inorganic sources, while others are heterotrophs, relying on organic compounds. Metabolic pathways include glycolysis, fermentation, and respiration. Bacteria can use oxygen aerobic or survive without it anaerobic. This metabolic flexibility allows bacteria to colonize a wide range of environments, from soil to the human gut. Bacteria generate energy through various processes. Aerobic bacteria use oxygen to produce ATP via respiration, while anaerobic bacteria rely on fermentation or anaerobic respiration. Some bacteria can even use light or inorganic compounds for energy. The efficiency and adaptability of these energy-generating mechanisms are key to bacterial survival and growth, especially in challenging environments. Bacteria reproduce primarily through binary fission, a simple process where the cell divides into two identical daughter cells. Growth rates depend on environmental conditions, nutrient availability, and genetic factors. Some bacteria can form spores to survive harsh conditions. Understanding bacterial growth and division is crucial for fields like microbiology, medicine, and biotechnology, as it impacts infection rates and industrial processes. Bacteria are masters of adaptation. They can adjust their physiology to survive extreme temperatures, pH levels, and salinity. Some bacteria produce protective structures like capsules or endospores. Others alter their metabolic pathways to utilize available resources. These adaptations enable bacteria to thrive in diverse habitats, from hot springs to deep-sea vents, and even inside living organisms. Any bacteria are capable of movement, thanks to structures like flagella, pili, or even gliding mechanisms. Flagella are whip-like appendages that rotate to propel the cell. Pili can help bacteria move across surfaces or attach to hosts. Motility is essential for finding nutrients, escaping harmful environments, and colonizing new areas. It's a key aspect of bacterial physiology and behavior. Bacteria communicate with each other through a process called quorum sensing. They release signaling molecules that allow them to coordinate behavior, such as biofilm formation or virulence. This communication is vital for survival, as it helps bacteria respond collectively to environmental changes. Understanding bacterial communication opens doors to new treatments and biotechnological applications. Biofilms are communities of bacteria that adhere to surfaces and produce a protective matrix. This matrix shields bacteria from environmental stress and antibiotics. Biofilms are found in natural environments, industrial settings, and medical devices. Their formation is a complex process involving communication, adhesion, and metabolic changes. Biofilms pose challenges in healthcare due to their resistance to treatment. Bacteria acquire nutrients through various transport mechanisms. These include passive diffusion, facilitated diffusion, and active transport. Some bacteria can even secrete enzymes to break down complex molecules outside the cell. Efficient nutrient uptake is essential for growth and survival, especially in competitive environments. The ability to utilize diverse nutrients gives bacteria an edge in many ecosystems. Bacteria must remove waste products to maintain cellular health. They use transport proteins and membrane channels to expel toxic substances. Some bacteria can convert waste into useful byproducts such as gases or acids. Efficient waste removal is crucial for survival, especially in environments with limited resources. This process also impacts the surrounding ecosystem, as bacterial waste can influence nutrient cycle. Bacteria encounter various stresses such as temperature shifts, toxins, or immune attacks. They respond by activating stress response genes, producing protective proteins, or forming spores. These responses help bacteria survive and recover from adverse conditions. Studying bacterial stress responses is important for understanding infection dynamics and developing new therapies. Antibiotic resistance is a major concern in bacterial physiology. Bacteria can acquire resistance genes through mutation or horizontal gene transfer. They may produce enzymes that degrade antibiotics or alter their cell wall to prevent drug entry. Understanding the mechanisms of resistance is crucial for developing effective treatments and combating infectious diseases. Bacteria can exchange genetic material through horizontal gene transfer. This process includes transformation, transduction, and conjugation. It allows bacteria to rapidly acquire new traits such as antibiotic resistance or metabolic capabilities. Horizontal gene transfer is a driving force in bacterial evolution and adaptation, making them highly versatile organisms. Some bacteria possess virulence factors that enable them to cause disease. These include toxins, adhesion molecules, and evasion strategies. Virulence factors help bacteria invade hosts, evade immune responses, and damage tissues. Understanding these factors is essential for developing vaccines and treatments against bacterial infections. Bacteria play vital roles in ecosystems. They decompose organic matter, recycle nutrients, and form symbiotic relationships with plants and animals. Some bacteria fix nitrogen, making it available for plants. Others help digest food in animal guts. The ecological impact of bacteria is immense, shaping the health and balance of natural environments. Bacterial physiology has many industrial applications. Bacteria are used in biotechnology, waste treatment, food production, and pharmaceuticals. Their metabolic capabilities enable the synthesis of valuable products, such as enzymes, antibiotics, and biofuels. Harnessing bacterial physiology is key to innovation and sustainability in various industries. Summary: bacterial physiology encompasses a wide range of processes that enable bacteria to survive, adapt, and thrive. From cell structure and metabolism to communication and ecological roles, bacteria are remarkable organisms with immense scientific and practical importance. As research advances, we continue to uncover new insights into their physiology, paving the way for medical breakthroughs and technological innovation. Thank you for joining us on this journey into the world of bacterial physiology.